Hello and welcome. This is Professor Rasmussen, Cognitive Neuroscience, episode number 5, the last installment of Cognitive Neuroscience. In a nutshell, this is going to be storing information. We're going to go from the small scale, from the neurons, to the large scale, to the locations of where information is stored. We're not going to get as in-depth as a biopsych class can, but we are going to get somewhat in-depth here. So, where do we start? Well, let's start with these two guys, David Hubel and Torsten Wiesel. Both of these individuals were super influential in mapping out the activity within an individual's brain, primarily non-humans. This is going to be a cat. Work with animals, as it says up there. And they worked on the visual system with these animals. Excuse me. Now, for their work, they won the Nobel Prize in 1981. Okay, so some very influential work. So what the researchers found is that upon implanting electrodes in various parts of the cat's brain, these cats were placed in a location where they couldn't move. This is unfortunate, but they could not move, and these electrical signals were implanted into these small areas within the cat's brain, and these cats were shown various stimuli that were on these computer type screens, some very crude computer type screens, and much of this work actually used overhead projectors. But anyhow, a stimulus was shown at a particular angle, and it actually took the researchers a long time to do this. They just didn't say, okay, stick that electrode there and move this little stimulus here and we'll get some activity. There were tons of failures that these researchers had, both within the area and to the actual stimuli. The stimuli oftentimes failed to produce early on activity. But anyhow, they finally got it to where they actually detected activity within the cerebral cortex, that's the outer layer of the brain, for the cat. When some shapes were put there, some edges were put there, and pretty much we're talking about an edge here. Okay, this is an edge. It's a line, but it's going to be an edge. It's going to be some sort of contour. And oftentimes what they also found is not only when the edge was stationary, but when the edge moved a little bit. So what we're looking at here are 2, 4, 5 different types of angles where lines where that line is going to move up and down. So the first one moves up and down. The second one is going to kind of move on somewhat of an angle. The third one is going to move on more of an angle. The fourth is even more of an angle. And the fifth, though, is, less of an angle really, and kind of just moving side to side, left to right. On the little black line, those 5 little black lines that you see right there, that is time again. So time is starting from 0 and moving on to the right, going all the way to, probably counting by milliseconds as well. Oh, I am sorry. I bet you guys are yawning now too. So anyhow, moving on down, you can see that these large black lines that kind of look like, it almost looks like as though it's a Richter scale for an earthquake. And you probably remember the Richter scale is if there's an earthquake, those lines that are typically just going straight across when the ground is flat are going to be going up and down because that seismograph is moving around. So that pen or pencil is moving around, and that's going to create this, the display of, the seismic waves, the earthquake. And that's exactly what's going on here. Those lines going up and down indicates neural activation. So what you can see that's really cool here is that these particular neurons, this is one particular neuron within the cat's visual cortex, that is the occipital lobe. What you can see is that in a particular manner at which this neural signal, the particular orientation and movement of a line, of a contour, of an edge, actually creates activity within the neuron. So you can see the most activity for this little neuron that this information is being recorded from is highly active for this third one here, when the line is shaped at possibly maybe a 45-degree angle, and it moves kind of on an upward right and a downward left slope. There's going to be a lot of activity. There's less activity when the line is placed a little bit more parallel to a 180-degree flat line. And there's a little bit less activity for the fourth one here, where the line is more, straight up and down. I guess you'd say that'd be like a 90-degree angle, perpendicular completely to a flat surface. Okay, so there's less activity. And when it's a flat line, whether it's going, just straight down, or when it's a flat line going up and down, the flat line going, left to right for the first one, and then the fifth one, there's less activity overall. There's very little activity in this brain cell, in this region for those lines. So number 2 is creating a little bit of activity. 4 is also creating a little bit of activity, but number 3 is creating maximal activity within that location within the brain, suggesting that that part of the brain, that in the little specific cell, is helping to code for possible movements such as what the cat is actually looking at. Now, if you put those together, the brain in the rear of the brain, the visual cortex, the occipital area, is going to have multiple different sites in each location, rather, that is going to be able to code for each individual movement. So this stuff is very, very complex within the rear of the brain, but it does look like small individual neurons are actually going to be coding for specific types of information, especially in the rear of the cortex, okay, for the occipital lobe. So how does that work when we're actually looking at people's faces? Well, this is going to be in a different area. All right, it's not necessarily going to be straight in the occipital lobe, but it'll be an area where vision is being processed. And so how about this person? Yes, we know who this person is, Halle Berry, right? So is it one cell? Here's a big kind of thought here. Is it one cell that codes for the comprehension and pretty much the recognition of Halle Berry? So this is called the grandmother cell approach, or the grandmother cell, hypothesis, also called sparse coding. Sparse coding means that there's very, very few cells, maybe even just one. Or is it many cells? Many cells meaning that there's multiple cells involved with your comprehension and recognition of Halle Berry, also called distributed coding. So let's take a look at this. So first of all, the grandmother approach. Okay, grandmother approach meaning that there's one neuron in your brain that is going to code for grandma. And when you see your grandma, that neuron's going to be active. If you're looking at another grandma, well, your grandma neuron is not going to be active because you're not mistaking that other old lady for your grandma. Okay, so the grandmother cell. Well, basically what's going on here is you just look at really like 3 neurons, okay? Or you can look at, a few neurons, 5, 10 neurons. But basically what you're going to see is that when you look at one person's face and you're like, oh, it's Bill. I know Bill. He's the bartender over at the, at the 5 and 9 or something like that. I don't know what the heck that place is. But anyhow, he's whoever. Well, that neuron 1 that you're recording from is going to have a lot of activity. Neuron 2 and 3, it has no activity. Okay? Okay, we'll get to it. Yes. Mary. Mary, she's that wonderful clinical psychologist who I met just down the road. Well, Mary, when I recognize Mary, boom, that's neuron 2. And then Ramon, he's a magnificent lecturer over at Northwestern. And Ramon, he's, his activity, or not his activity, but when I see Ramon and I recognize Ramon, boom, that's neuron number 3 coming online, okay? So this would be the sparse coding, grandmother cell, single cell recording, to suggest that one neuron is regarded in the recognition of these individual faces. Now, there are some potential problems if the brain works like this, okay? So think about that. What happens if there's cell death? And yes, cells do die. It is possible that a grandmother cell could die, and therefore if a grandmother cell died, you wouldn't be able to recognize Bill, Mary, or Ramon, and even worse, if the grandmother cell was attached to, like your grandma, encoded for your grandma, well, you wouldn't be able to recognize your grandma, okay? So another problem is that you might run out of neurons, right? Like, how many faces do you know? You know a lot of faces. I know a lot of faces. Names is a different story. Names are a different story. But faces, pretty good at recognizing faces. So, if we know, thousands of faces, well, we're going to need thousands of cells. Well, we're probably not going to run out. We got millions upon millions of cells, but it's possible. All right? Now, distributed coding looks like this. It's a pattern of activity, okay? It's not just one neuron or just a couple. It's hundreds to thousands to definitely more like thousands to millions. So, here's the deal. For Bill, that bartender over at, Goodfellas or Good Boys, whatever that bar you go to, and get your little burgers at, because most of you aren't 21. Bill, his neuron, or your recognition of Bill is activity of both 1, 2, and 3. A lot of activity in number 1, a little bit in number 2, and then minimal in number 3. Old Mary, hers is a little bit of activity in number 1, a lot of activity in number 2, and a lot of activity in number 3. And that particular sequence that you have activity for is going to chime into your brain, that pattern of activity, that is Mary. I'm looking at Mary. Ramon is a lot of activity, little activity, and a lot of activity. And now we can even throw in, the cool thing here is we can throw in Roger. Roger, you know this guy. He's that student that you know. And Roger, with 3 neurons, you can actually have the ability to detect another individual with just 3 neurons. So Roger is a little bit of activity, very small amount of activity, and then a little bit more of activity for neuron 2, and then a pretty decent amount of activity for number 3. But that would be Roger's pattern of activity. So you can see that what this allows for is an individual can code for more faces, recognize more faces with, yes, the same amount of neurons. So 3 neurons can now get away with being able to detect many different faces, and it's all the pattern of activity. Now, could have given you the numbers earlier, but nonetheless, there you go. You got it, okay? So basically, the interesting thing is, if a cell dies, it may not mean as much for the recognition of an individual. You still might not be able to recognize the individual exactly, but it would be better in that, it's just one neuron, and there's these other neurons that are taking place, and you'd likely be able to say, oh yeah, from this pattern of activity, yes, that's Roger, or that's Mary. Okay? It doesn't rely so much on just that one-to-one coding, one face to one neuron. Are you going to run out of neurons here? Well, it's probably less likely that you run out of neurons because 3 neurons can code for many faces, hundreds of potential faces, depending on the pattern of activity. Now, there actually is some research for some of these types of debates. This question is the grandmother of the cell versus the distributed coding. Well, actually, cool enough, is looking at patients who suffer from seizure disorders, again, electrodes implanted into their skull. What researchers have found is some pretty decent evidence that in some locations for an individual and for some particular faces that there actually may be more of a type of thing of like a grandmother cell. Interestingly enough, it looks as though there's a small amount of neurons, maybe tens to hundreds of neurons, maybe even single neurons that actually code for particular faces. So there are particular neurons that look like they code for people like Steve Carell, the funny actor. I think he's funny. Some people think he's stupid, like, well, my wife and my daughter. Some people whose, opinion is just, who knows? But anyhow, Steve Carell, he's a funny guy. And pattern of activity in these single cells is actually quite substantial, that when an individual looks at Steve Carell and they know who it is, there's likely going to be one or very, very few amounts of neurons that are going to be active. Same even goes for others. When other people are showed, so these are people who are suffering from a seizure disorder, who had these electrodes implanted in parts of their temporal lobe, and they were shown pictures of people as well as objects. There was a particular amount of activity depending on the face that they were looking at in some very, very small and very, very specific locations, suggesting that down to just like maybe the one neuron to the small tens to 20 neurons that are coding for an individual, such as the individuals you're looking at. Interestingly enough, it wasn't just for people, but also various places. Say places in France, the Eiffel Tower, Tower of Pisa, Italy, various structures that are very, very famous and well known. Okay, so let's move on past the coding debate, and this is still debated today, whether it's sparse coding or grandmother coding, and let's actually look at the pattern of activity within the brain. Okay, so this is a really cool research that actually comes from Carnegie Mellon, however you want to say it, but Carnegie Mellon, and many people pronounce it differently. It's a university out east, and very, very good cognitive psychology program, and there's these researchers, primarily Marcel Just. Okay, he's one of the authors here, one of the last authors, but nonetheless very large in the cognitive psychology world. And what they looked at is fMRI. Okay, so functional magnetic resonance imaging, looking at the blood flow within the brain during a actual cognitive processing task. Okay, where these people had to look at two different types of objects. They had to look at either a dwelling, which is like some sort of home type location, or a tool. Okay, so thinking of homes versus tools, and not just thinking of them, but actually seeing them and having to register what these objects were. Now, these individuals were in an fMRI scanner while they viewed these images on a computer monitor. Now, the computer monitor was detecting the neural activity while these individuals were doing their task of looking at either the tools or the dwellings. So basically, what happened here is these people saw 10 pictures while in the fMRI. Okay, and they're asked to think about the properties in each of these 10 pictures. Okay, the tools versus the dwellings. Okay, the participants then came back later on to the fMRI, and the computer actually tried to guess which picture the person was actually looking at. So originally, the individuals were just sitting there, not looking at anything, and then say a tool came on the screen. The person was instructed to just think about the properties of a tool. A blank screen would come on, and then a dwelling might come onto the screen for a number of seconds, and then the person was to think about what the dwelling was, so on and so forth, the aspect of it. All the while, the computer is recording the activity within the brain. Okay? So now, the participants after that series, now they come back to the fMRI, and the computer has to try to guess which picture the person is now looking at, whether it's a tool or a dwelling. Okay, so this is pretty cool stuff. So this is basically kind of like almost getting at like a lie detector, but there's no line involved. It's almost like a mind reader. Okay, so this is super cool research that Justin and colleagues were looking at here, especially Shinkara. Okay, sorry about that. Shinkara. I have not pronounced your name in quite some time. But anyhow, what these researchers looked at is basically the activation when they were looking at those pictures compared to what the computer gathered from this beginning, from the baseline. And the baseline is when the individuals looked at the pictures to begin with. So they're comparing what the people are seeing now to the baseline when they actually were looking at the pictures in the beginning to kind of try to read the participant's mind. Now there's two different analyses. The first one is for the baseline for the participant, and so this would be called a within design, where an individual, the researchers are looking at how does the data compare now upon the second trial when the participant came back in to parts of the first for that participant. Okay. Now the researchers also compared the baseline overall from the group. All right, and this would be the between subjects design, to where the researchers just look at, okay, overall, when participants look at this area, in general in the baseline, for this particular picture, what area is becoming active, and then we'll kind of compare that to what this individual is doing when they're looking at these pictures, and we'll try to guess what that individual is doing. So, here is kind of how it looked. Participants would look at the tool for 3 seconds, think about the properties, and then a blank screen with a fixation point would pop up for 7 seconds, a dwelling, fixation point for 7, a tool, fixation point, and then it just happened to be a tool, so on and so forth. Now what you're going to see is the within participants versus the across, that is the between participants. Here this is only 11 participants, so a small sample size, but this fMRI work is quite detailed and time sensitive and actually quite expensive, so a small sample size, but nonetheless. Over here you have the classification accuracy of the computer, so how accurate the computer was based on a 0 to 1 scale, 1.0 scale, 1.0 being like 100% accuracy, 0.9 would be 90% accuracy, so on and so forth. And so what you see overall is, just in general, 12 participants. In general, within accuracy, it ranged at about 87%. Okay, that was from their first trial to their second trial in the fMRI. Now the 12 participants in the between subjects design, that's when you start to group this information together for these individuals. It dropped down a little bit, but to 82%. And this is, again, fairly good accuracy for both of these, for the within and the between. Now, let me show you something. I'm going to have to exit out of here really quickly. Okay, sorry about that, folks, but let's go back, and I want to show you the actual data. I'm not sure why that didn't pop up, but here we go. So you can see the black line is essentially for participant number 4, 8, 6, and the white line is the cross subjects analysis, and you can see that for 4, 8, 6, so on and so forth. And overall, what you see is the 0.6 would be at the rate at which, 0.5 would be at chance, if the computer was just guessing what the participant was looking at. So let's pretend participant number 4 is, in one particular situation, they're looking at a tool. Well, the computer has 50% accuracy, whether it's, it's a tool or a dwelling. So 0.6 is about the point where if it's higher than 0.6, if this average, across those 10 trials is higher than 0.6, well, then the computer is actually better than average at a rate where it wasn't probably just caused by chance. The computers actually can detect what the person is looking at, not at a good rate. Okay, just being a little bit above chance does not mean the computer is that accurate, but it's more accurate than just guessing. Okay, and so that's the cutoff here. And you can see that overall the rates of detection by the computer are actually quite high. Especially for some participants, 4, 8, 6, so on and so forth. Participant 1 and 3 kind of get a little bit lower, but overall accuracy is quite high, especially for the within. The accuracy drops a little bit, especially in some of these individuals, P9 and P5. Some of these individuals were quite low for across subjects, but overall you can see that, as we said just a moment ago, it's still quite high. So 87% for the black lines and 82% for the white lines, suggesting that the computer is really, really good at detecting where a person is, what a person is looking at, first off, and then be able to basically read a person's mind from what that person is looking at. So this is interesting. So what's going on here? Well, basically, the computer seems to know this because when you look at a tool, there's going to be a particular level of activity in a certain area of the brain, and when you look at a dwelling at a home, there's going to be a particular amount of activity within a particular portion of the brain, suggesting that when you look at something, there's going to be a specific activity. When you look at something else, there's going to be a different level of activity, suggesting that these things also are not just the same for you. So like when I look at a tool, the same part of my brain is becoming active, active, active. Great. When I look at a dwelling at a home, active, active, active. This is stable across participants, so that when I look at a home, at a dwelling, your brain also is becoming active in a similar location when you look at a dwelling. When you look at a tool, well, a particular part of my brain that is typically the similar area that is active in your brain when you're looking at a tool is going to be active in my brain. So this is kind of cool stuff, looking at the shared similarities between where these objects are actually being processed within the brain. So this is the take-home message. The activation of information, tools, and memories about tools is distributed throughout the brain. However, the pattern of activity is similar amongst humans, and that the computers can detect this. This is really suggesting that it is quite similar through humans. Okay, so guys and girls, what we finished there was the cognitive neuroscience part number 3, the 5th one in the whole series, storing information. Hopefully this has been helpful on the neuron level, all right, the neuronal level, looking at the individual neurons, to the more global level, looking at the locations within the brain where this knowledge actually comes from. Guys, girls, it's been a pleasure. Thanks for tuning in.